USGS Science for a Changing World, ORDA Science Webinars. The Office of Restoration and Damage Assessment hosts a monthly series of science webinars intended to provide our NRDAR practitioners, as well as our co-trustees, the most up-to-date information related to NRDAR practices, as well as cutting-edge science topics related to the field. Typically hosted by Jo Ellen Hink, USGS National NRDAR Coordinator, these webinars are available as recordings after the fact and posted on ORDA's website. For more information or to suggest any future topics, please contact DOI NRDAR at ios.doi.gov. ORDA Science Webinar, Contemporary Data Analysis. Jo Ellen Hink, Moderator.
I just tried to push that button and it doesn't look. Yeah.
There it goes. Okay, perfect. Sorry.
All right. So we're recording, everyone. So if you don't want to be recorded, now's the time to pop off. We'll take questions at the end of the webinar, and you can use the chat feature, which is the little bubble up at the top of your screen if you're not used to Teams. If you're having connectivity problems, then you can make sure that your camera's off, and please make sure that you're on mute during the webinar. So we have our disclaimer as well on the screen for that our lawyers like us to put on. So I'm going to go to the next slide. Just for an announcement of upcoming webinars, we have 3 in queue. We're going to take a little break in August. And then in September, one of our solicitors will be talking about, do I need more science to build my claim? When is there enough science? A scientist would say never, but sometimes for NRDA, the answer is yes, we have enough. And so we'll be learning a bit about that. And then in October, we're going to be looking at oil impacts on bird migration. Some of this work started back in Deepwater Horizon and has continued. And so that should be exciting to learn about. And then getting to our 101 topic series in November, we're going to have Ron McCormick from BLM talking about developing a conceptual site model. So with that, I'm going to stop sharing my screen and hand it over to Richie and Barnett.
So thank you for the introduction and just the brief overview and things. So as we get started today, Barnett and I will be going back and forth. So to get started with our introductions and things, who are we and why are we here? My name is Richie Erickson, and I'm a research quantitative ecologist at the Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. Primarily, I model invasive species. So I lead the Department of Interior carp population modeling efforts and also do environmental DNA modeling. But I have other duties as assigned. So I serve as a statistical consultant at my center and also do statistical reviews. Also, last, I'm an editorial board member of Environmental Toxicology and Chemistry, so I see a lot of different ecotox studies. In these last 3 sort of roles, I've saw a lot of people wanting to move beyond p-values, but not know options. So Barnett and I worked together on a paper that led to this talk as well. So Barnett.
Hi, it's great to be here today. I conduct lab, pen, and field exposure and effects studies, mostly in wild birds and wild mammals, examining molecular through population level effects. I've done work with persistent organic pollutants, metals, insecticides, and most recently, rodenticides. I've been involved in several natural resource damage assessment studies in the past decade. And historically, I contributed to the biological resource injury definitions that appeared in 1986 in the Federal Register that were described under natural resource damage assessments. Okay, Richie.
So to get started, we just have some statistical quotes to get your minds thinking. So one I like is from Ernest Rutherford, a famous Nobel laureate from chemistry. If you need to use statistics, then you should design a better experiment. From Stephen Colbert from the Colbert Report, equations are the devil's sentences. Carl Pearson, a famous statistician, has noted that statistics is the grammar of science. Barnett has a quote he wants to share as well.
Yeah, in the Complete Idiot's Guide, statistics plays a vital role in today's society by providing the foundation for sound decisions.
Robert A. Donnelly.
In the next slide, I would recommend reading a great publication I felt, Principles of Sound Ecotoxicology by Harris. It appeared in the journal Environmental Science and Technology in 2014. It provides a great description of some of the characteristics of robust studies, in particular, things like study planning and statistical design, replication and weight of evidence, all of which are germane to NRDAs. The next slide kind of shows the paper that Richie and I worked on and published as a critical perspective, moving beyond P is less than 0.05 in ecotoxicology, a guide for practitioners. And that appeared in Environmental Toxicology and Chemistry last year. And it's really the basis of that paper that led to this invitation to give a talk today. Okay, Richie.
Thanks, Barnett. And also, Joe shared a link to this paper in the chat. So if you want to get it, it's available there. So as we start off, we're going to talk about what are p-values, because most scientists don't know. And also, why do we need to change? Without knowing this, it doesn't make sense to look at the alternatives. So Michael Newman, who is retired from VIMS out of William and Mary, he's done workshops around the world, and he would ask people, when you took statistics or when first exposed to significance testing, you were taught, what is statistical significance? Then he would ask the participants, and he would give people a multiple choice question with these 6 possible answers where people could ask what it is. And I'll give everyone a few seconds if you're able to read along and just read through, and you can, in your head, just think what do you think the correct one is.
When you took statistics, or when you were first exposed to significance testing, you were taught the statistically significant outcome with a p-value of 0.5 means. 1. The null hypothesis is disproven. 2. The p-value is the estimated probability of the null hypothesis being true. 3. You've proven that an effect is present. 4. You can deduce the probability of the effect actually being present. 5. You know the probability that you would make a wrong decision if you rejected the null hypothesis. 6. You know the probability of getting these, or more extreme, data if the null hypothesis were true. 7. If you repeated the test many times, you would obtain a significant effect in 9 to 5 out of 100 trials.
So the correct answer is actually number 6, but most people didn't know. So Mike's done workshops around the world, everywhere from his home institute, Virginia Institute of Marine Sciences, Savannah River Ecology Lab, and several in Asia. And most people got this answer wrong. In fact, the average response correct rate is worse.
I think Richie froze, and maybe he'll turn his camera off.
Yes, Barnett, this is Joe. I could hear you speak, but
chance alone.
Oh, here he is.
Yeah, Richie, turn your camera off.
My camera is off. Can you hear me or not?
Yes.
Okay, sorry about that. I'll make sure yeah. So this is the informal definition of a p-value from the American Statistical Association, and it's just a nice working definition of what it is. So everyone's had a chance to read it. So
you know the probability of getting these, or more extreme, data if the null hypothesis were true.
But what are the problems with p-values? Like, why do we need to move on? As a silly example, there's a webcomic called XKCD that I really like, and it's just a neat comic, and they publish under an open access license, so we can include their comic here. So imagine somebody comes running to us as scientists, maybe it's the NRDAR lawyers, saying, we hear jelly beans cause acne. So they say, scientists investigate. And the guy's like, the scientist in the background's like, but I want to play my Minecraft. Fine, I'll go ahead. So we do a study. We found no link between jelly beans and acne, p greater than 0.05. But maybe it's actually the color of the jelly bean that causes the cancer, or causes acne, I'm sorry. So we repeat it with 20 different jelly bean colors. And we happen to find that 1 out of 20 happens to be linked with acne. So hyping science, as it's often done today, we run out. Maybe our university's press office would say, it settles it. It's only certain colors that cause acne. So we get the headline, green jelly beans linked to acne. So it's a silly example of how statistics could be misused. But this is actually a broader problem in science. These are example headlines that show how there's a huge replicability crisis going on.
Headlines: Replication crisis spurs reforms in how science studies are done. The statistical crisis in science.
In the biomedical field, it's billions are spent on irreproducible results. And just these examples share how it's a big problem. And problems such as this have caught the attention of the American Statistical Association. So in 2016, they issued their first ever policy statement, the ASA's Statement on P-Values, Context, Processes, and Purpose. So this was just trying to define how to correctly use p-values and make sure they weren't incorrectly used. And then they, however, followed up with another editorial in 2019 in a special issue of the American Statistician talking about moving to a world beyond p-values. So we'll talk about more of these in detail, but this is just showing that the misuse of p-values is large enough the American Statistical Association is taking some action on it to try to get people to use more appropriate statistics. So their 2016 principles include looking at what p-values can indicate. They can indicate how incompatible the data are with the specified statistical model. P-values do not measure the probability that the study hypothesis is true or the probability that the data were produced by random chance alone. This is one that's especially of interest to this group, especially with decisions. Scientific conclusions and business or policy decisions should not be based only on whether a p-value passes a specific threshold. Proper inference requires full reporting and transparency. A p-value or statistical significance does not measure the size of an effect or the importance of a result. Last, by itself, a p-value does not provide a good measure of evidence regarding a model or hypothesis. So, as you can see, the American Statistical Association's sort of defined what a p-value is, and their white paper and policy statement goes into greater detail about these.
So now a little bit about use of the word significant and the phrase statistically significant. The next slide, in 2019, Wasserstein and colleagues in an editorial came right out and said it's time to stop using the term statistically significant entirely, nor should variance such as significantly different, p is less than 0.05, and non-significant survive, whether expressed in words, by asterisks in a table, or in some other way. On to slide 23, and this proposal is actually still being debated a bit. It's a work in progress.
Title: The P-Value Statement, 5 years on.
So by example, in slide 24, using some of my own data, one could say blood clotting time was significantly different in warfarin-treated kestrels compared to controls. P is less than 0.05. But it would really be much better to say blood clotting time was prolonged in warfarin-treated kestrels, prolonged indicating the direction it was affected, compared to controls. And when we compare it to controls, we come right out and state the data were log transformed. We used a one-way analysis of variance. Here's the test statistic. Here's the exact p-value. We did a multiple comparison procedure. The groups were different by the control by values of less than 0.0045. And the best thing, in my opinion, is we show as much of the data as we can in a figure. Richie, go on.
Thanks, Barnett. Sorry, I have a 5-month-old baby who's upset with nap time in the background. So sorry if you hear that. So what are alternatives to p-values or null hypothesis significance testing? I mean, we said not to use them, but what's better? So in the same editorial that we've cited before, there's this good quote that what you will not find is one solution that majestically replaces the outsized role that statistical significance has come to play. So there's no single silver bullet. Instead, what are some things we can look at? We can correctly use the null hypothesis and describe the results. We'll provide an example of this. You can look at the effect size estimation. So rather than looking at like the p-value, what comes out of it? We'll provide an example of this. Also, how uncertain, what's the uncertainty and the confidence and credible intervals around it, and things such as reference intervals or detection limits. Model selection, so AIC or other approaches where you have multiple competing hypotheses. There is appropriate hybrid methods. There's also things such as structural equation modeling and Bayesian belief networks. So those are some of the many examples. So
and now some of the examples. Let's go to the next slide.
Whisker chart example, blood HG in loons. Chart displays mercury level boxes for female and male loons, with whiskers extending beyond the boxes.
In my opinion, presenting data in a figure or drawing is often worth more than 1,000 words. This is a box and whisker plot showing the extreme lower value, the first quartile, the median, the third quartile value, and the upper extreme value. Is the difference important? Richie, go on.
So with the loon example, male loons had higher average blood mercury concentration than female loons. So this is how you might report it. The first part is the point estimate. So it was 0.263 micrograms per gram. Next, we report the uncertainty. So what's the 95% confidence interval? Notice it doesn't include 0. If we want, we can still include a t-test and all the appropriate results. So this includes the p-value, exactly, the test statistic, so the t-value, as well as the degrees of freedom. But as the statistical person here, I might say, so is this important? And that's where I hand off to Barnett, who is the biologist.
Supposedly, yes. And what I guess I would say, are these data statistically important? They sort of demonstrate that mercury is transferred from the hen into its egg, and that maybe accounts for the difference in concentrations between male and female birds. There can be other factors involved. How about from a hazard or risk standpoint? Well, oftentimes, unfortunately, there's no simple line in the sand. The mercury threshold affecting reproduction in loons is about 3 micrograms per gram wet weight in blood, as reported by Dave Evers. And then there was a great meta-analysis, several done, one by Phyllis Fuchsman in 2016, and she found pretty close to what David reported, with an EC20, an effect concentration 20 on reproduction, at about 4.3 micrograms per gram wet weight in blood for larger size birds. While others, Josh Ackerman and Allison Jackson, they suggested that the effect threshold on reproduction in fish-eating birds from a moderate risk standpoint was lower, about 1 microgram per gram. So let's go on to the next slide and say a little bit about the use of reference intervals. This was introduced in human and veterinary medicine around 1969, and many have found that it is of application and use in ecological risk assessment, and it may also be of use in natural resource damage assessment.
Cluster graph sample, Graph showing prothrombin time in seconds on the left axis. Clusters are bald eagles, Cooper's hawks, and red-tailed hawks. Most of the reference points fall between 8 and 17 seconds. A few samples fall far above the clusters at 80 to 100 seconds. A solid line runs across the graph at around 25 seconds.
By convention, it uses samples from at least 20 healthy, unexposed individuals to come up with the range of values or the interval that encompasses 95% of the healthy population. So here, for the American kestrel and blood clotting time, we have a reference interval that we reported based on 49 individuals, ranging from 8.3 to 16.5. While less than ideal, we have used this interval in American kestrels to examine clotting time data from other raptors, species like the bald eagle, Cooper's hawk, and red-tailed hawk. And in the next slide, the one you're on, here is this dark line, and that's actually 1.5 times the upper reference limit that we found in kestrels. And the reason we use that is we really didn't have a good reference interval for bald eagles or Cooper's hawk or red-tailed hawk. We did the best we could do. And perhaps the most important data in this slide are the values well above that 1.5 times the upper reference limit. These are the outliers. And these values, in combination with other data, things like pathology and residue concentrations, can help reveal what is going on sort of by a weight of evidence approach. And in fact, some of the values for prothrombin time that were very, very high, those birds also had high concentrations of anticoagulant rodenticides in their liver. Let's go on to the next slide.
Second cluster sample, limits of detection for selenium in loons. Left axis is blood selenium and bottom axis is mass. Data points are color-coded for female, male, and unknown gender.
These are some data on selenium concentration in blood of loons. And it's important to mention how values get handled that are below the detection limit. In this slide, there's a red line, and if you count the dots, there are 6 values that are below the detection limit. The investigators that published this paper assigned those 6 samples that had values below the detection limit at a level of 1/2 the detection limit for their analyses. Perhaps not the greatest idea is shown in the next slide. As pointed out by Dennis Halsell, substituting a value of 1/2 the detection limit, he considered it a perilous error. It can produce a false signal. There are many methods to handle such data. I like perhaps the simple approach, one of them using a non-parametric method, and then generating a mean. It's not a single value, but it's a range. That is a Kaplan-Meier mean. So you assign the non-detect values a number very close to 0 and calculate a mean. And then you assign the non-detect values a concentration very close to the lower detection limit and calculate a mean. And then you present the mean as a range of possible values. Richie, why don't you go on?
Sure. So moving on to another approach is model selection. And with this approach, you build competing models. So these models represent different hypotheses usually. For example, we'll provide some examples with the next slides. And then when you have the models, you compare them using a selection approach. Probably the most popular and famous one is AIC, but many other alternatives exist. So as a starting example from a textbook, which curve is best for a dose response curve? So we have 3 different curves.
Going left to right, the first curve runs in a smooth curve from top to bottom. The second curve is slightly flattened at the top and bottom, and the third curve is almost flat at the top and bottom with a steep descent between.
The curve on the left has a 2-parameter model that assumes a maximum of 1, a minimum of 0, So it just estimates how steep the line is and where the curve is at. The second model is a third 3-parameter model that has a different maximum value, but still a minimum of 0. The fourth model estimates a different maximum and a different minimum, as well as the slope and intercept. So this is a textbook example. And just looking at these, you can see that model 1 on the left should not be at 1. So it could be at model 3 or model 4. And then looking at the model selection, we look at the AIC results with the table. And the lower AIC score just means a better fit. And this shows us, looking at the weight of the model, that the 4 or 3-parameter models, LL3 and LL4, are similar choices. So if you just want to take the best model, you might use the 4-parameter model. You can also use model averaging, where you use the results from both models and then average them together. Or as a biologist, you might say the 3-parameter model is biologically tenable. Choose that one and justify that for your later work. So AIC lets you compare models that way. Next, Barnett's going to talk about an example he's used for real data.
Sure, and it's not actually competing models, but it's competing distributions used in generating toxicity reference values. That is a dose response curve from which an adverse effect threshold can be described. And this is an example of a preliminary toxicity reference value describing the dietary concentration of chlorphacinone that would have to be consumed by an American kestrel to prolong clotting time. So looking at this graph, at 26 micrograms per kilogram body weight per day, about 10% of the exposed birds or population has prolonged clotting time. At 39 micrograms per day, about 50% of the population has prolonged clotting time. And at 52 micrograms per kilogram body weight per day, nearly all of the exposed birds have prolonged clotting time. Generating toxicity reference values requires a lot of data, and it's best to have data from 3 different species in a vertebrate class representing 2 different phylogenetic orders, 2 chronic lowest observed adverse effect values, and 1 chronic no observed adverse effect level value. It's a tough requirement to get all of these data, and I proposed this as a preliminary TRV and explained all the uncertainty and limitations, which is important to do for any data set. Richie, go ahead.
Thanks. Another example of how I've used model selection was looking to see where endangered bats live on the landscape, because we were interested in both white-nose syndrome and also wind turbines.
Cluster maps showing the habitats of endangered bats in the central and eastern United States.
Modeling their location, and we didn't have enough data to necessarily use all parameters and things, so we compared different hypotheses about what are important. So then we were able to create this map here and use the results with some other modeling and risk assessment work. From this, we then compared the different WAIC scores to see which model was best. And then we also reported the coefficients as well. So that's an example of how we've used a model selection for some of our own work. Next is another approach, which is called time to event analyses. So historically, these are known as survival analysis, and it's something that's becoming increasingly used with ecotoxicology and other events, where you look at something such as death or another event to see how long it takes. This example looked at how long catfish could survive when they're exposed to different levels of salt water. You can see below 12 parts per thousand is the top of the figure, and those individuals lived the entire duration of the experiment. But then as the concentration of salt water increased, the curves go farther to the left, so the catfish live for less time. And this can be done for any sort of toxicological endpoint or other endpoints as well. And it's also commonly used, for example, in engineering to model how long a part might last in your car until it dies. So that's just one example. Another example, as an alternative to just null hypothesis significance testing, are regression analyses. And regression could be its own year-long graduate level stats course. So in one slide, regression analysis, you have a model where you have y predicted by some coefficients beta or a vector, and then some data x. And if beta is different than 0, you say that x is associated with y. So there's assumed to be some important relationship. So as a statistician, that's something I could tell you there is the relationship there. And then Barnett or another ecotox person, the scientist, would help figure out what it means biologically. Things can also be corrected for. For example, medical studies, if you read the paper, they might say the drug corrected for this, like age or sex or something, does something. So that's how you include multiple covariates, and that's where that term comes from. There's also many different flavors of regression. There's generalized linear models, like a logistic regression, generalized additive models, things such as splines, hierarchical or mixed effect models. There's a lot of names. Also, it's worth noting that an ANOVA and even a t-test are both special cases of a regression. So regressions can be used with p-values, but they can also be used without p-values. Moving on, another way to do this and move beyond p-values is looking at the effect of things, but also adding context. So for example, let's say Barnett does a field study with ospreys and finds a 10% decrease in recruitment. What does this mean in the bigger picture? Does it matter, or is it a trivial decline that doesn't matter? For example, when I was working with the USGS's Wind Energy Impacts Project, we looked at things like this for songbirds versus eagles. And a songbird that's short-lived might not be impacted by a 10% decrease, but something like an eagle that's very long-lived and has few offspring would be. And to further quantify this, we can use the existing knowledge of the system and things such as population modeling. This is currently commonly done in ecotoxicology, and Valerie Forbes has done a lot of work, such as the example paper I cite here. Also, with ecological risk assessment and EPA's registration process, they commonly use population models with that as well. So there's precedent there if you're looking for other people using this approach. Another example, and I think this actually ties into your October talk.
Infographic, energetic balance in oiled birds. Graphic displays how oiling can affect food consumption, thermoregulation, and migration.
Wayne Thogmartin and Mike Hooper, who I've worked both with before, and others are doing a population model where they're looking at energetics of birds. So they have a bird model, then they look at how energy gets changed from different stressors and toxicants. And then this can look at the impact across the landscape. So for example, a bird that spends the summer in Wisconsin and the winter in Louisiana, how does it get impacted by an oil spill in Louisiana or something? And then there's lots of other uses as well in energetics. So if anybody, we cite paper examples in our paper, and I can also share some links if anybody wants to know examples of this or other examples. So another approach are structural equation modeling. These construct causal diagrams. Jim Grace is a USGS expert on the topic. He actually wrote the ecological structural equation modeling book, and he's out of Louisiana. For an example, with my own work, we were looking at what predicts environmental DNA in a lake, and we just constructed different diagrams. So we looked at how depth and the hard surface affected the ash-free dry weight. So they actually went out and got the zebra mussels and sampled them and brought them back to the lab to get their weight. And then we looked at how that related to environmental DNA. I also, just with this example, need to slap myself on the wrist because I was misusing statistical significance in the caption and things. So
causal diagram samples. The studied aspect and numeric results progress left to right.
It's a big change for all of us, and that's just another small take home from this slide. Another topic is Bayesian belief networks. I know that there's an RFP out for this, and you've had Wayne Landis talk about the topic.
Web chart sample. Main subject is island elevation. Cross-related subjects include mean recharge, island width, water table max, and mean depth to water.
So compared to a structural equation modeling, it's more focused on correlation rather than cause, but both are closely related. For example, Mike Feenan's a hydrologist I work with, and he just had a different example of using a Bayesian belief network to simplify a complex hydrological model. so they can construct something here and then use it to inform water management. So this is the end of our specific examples, but just for some broad general guidance, the first sort of important thing is to define what is important. So if you're a scientist, what is your hypothesis? If you're looking at risk and impact, what's the important damage? And this is hopefully where us as scientists talk to the risk people like the lawyers and just understand the system. What's important? What do we want to show? Then next step is just get the data to show what's important. This might be doing, I know, like NRDA, you fund your own studies, or maybe there's existing data out there as well. And then once you figured out what's important, estimate it. And that's where statistics comes in. What's the uncertainty and what's going on? And uncertainty is just a major point, because if you look only at p greater than or less than 5, you're missing capturing that uncertainty. Also, the American Statistical Association included some of these things. So one is the acronym ATOM, and that's accept uncertainty, be thoughtful, open, and modest. So accepting uncertainty is doing things like reporting your estimate and the uncertainty around it. So not just your mean, but your confidence interval. And the thoughtful, open, and modest. So realize that your results fit into a bigger perspective. So be thoughtful and use like a weight of evidence. No single study reinvents anything. Really, it's repeatability and just being open with your method so other people can repeat what you did, and also have access to your data and other things. So a lot of the open part, USGS already does, but then it's also, there's a big problem where scientists don't report p-values unless they're significant. So for example, psychology has had a replication crisis because there's been a lot of studies where people can't recreate other results. For example, like the people where they test college students for different things and they find these weird results. Turns out they don't hold up with replication. Last is the modest, and that's, no single study presents all the solutions. So that's like the weight of evidence idea. And then just some other guiding principles are things like there's no line in the sand. Think about how things are uncertain. And also last, think about why. Even if you find weird results, what's going on with them? And or if they're not weird, how does it fit into the existing theory and what's the process? So for the last slide, we know NRDA is interested in moving on possibly to things besides p-values. So one thing is things such as study plans. And if you're going to use these, include the proposed statistics. This is something that I do, for example, at my center. Almost all the studies have review plans that get reviewed, and I review them. So it's an area where USGS could help if you want. Just having reproducible results and workflows. This is also another topic for a different day, but people need to be able to recreate your statistical results and your workflows, and the ASA has published another policy statement on this. It also ties into things such as quality management system, QMS, that USGS is moving towards, but for statistics rather than lab data. So for example, if you use GLP in the lab, you want to have reproducible results when you're out of the lab as well. Another important thing is matching your study design to your statistical methods. It's just really important to know what's going on and have something well designed to match it rather than try to shoehorn one into the other. I would also add, for example, I have a master's student who we've submitted her paper to under review, but we looked at some of the OECD guidelines and found that they're outdated for how they suggest analyzing results. So be aware of new statistical methods as well for your studies, and things that are 20 years old might recommend methods based upon old computing limitations that are no longer important. Another way we could help is comparing statistical approaches. Maybe you're concerned that the new approaches won't be tenable or something, or you don't know how to describe them to existing approaches, or just curious what is the difference. That's another way that Burnett and I could help. And last, we're open to other ideas. We want to hear what you think is important and what you're worried about for things. So that's the end of our talk. We have plenty of time for questions, and this is a link of resources as well. We'll leave up on the screen for a little bit.
Resources can be found in the PDF in presentation details or follow https://bit.ly/3 lowercase v, lowercase h, capital W, lowercase z, capital Z, capital D.
Well, Richie and Barnett, thank you very much. I encourage folks to type their question in the chat, or if you would prefer to raise your hand, I can monitor that and you can come off mute if that's easier. As we're waiting for folks to do that, I'd like to get your all's take on John Eisenhardt's comment in the chat that he noted that the CERCLA NRDA regulations use the word statistically significant 14 times. Yes, and before we go any further, just a reminder that this webinar is open to the public. That's the friendly reminder from your solicitor's office in DOI. So Barnett and Richie, do you have any thoughts on where you have this language in the regulations versus some advancements in the science?
Sure. Well, I'll make a quick comment. And it was very astute of John to point that out. And I'll point out that I believe those guidelines are now 35 years old. So maybe some updating or slight modification is in order. John will correct me if I'm wrong on the age of those guidelines.
So one other thing I have to add to that is, even if you're tied to using statistical significance, look at Barnett's example he shared, where his first example was only the p-value, and you say it's significantly different, whereas his second example showed the uncertainty and all the estimates. So even if you're tied to the statistical significance, still report how variable things are and what's the uncertainty and the effect, not just the p-value. So that's my suggestions. Barnett has a good one about the age of the regulations. The other is looking at, if you are within that framework, look at how report what you're reporting, not just the p-value. So, John, do you have anything you want to add?
No, I appreciate your comments. It's I guess full disclosure, Richie and I talked a month ago or so about some of these topics that were presented today. And it's had me thinking for a while about some of these issues. For those of you familiar with at least the CERCLA NRDAR regulations, that's mostly mentioned in the sections that have to do with methods for demonstrating injury. So that's, if you're looking for that in the regulations, obviously you can search it in the document or go to those subsections. So I think, I'll just say that I think that the comment about maybe some updating, or at least looking at how we're studying or investigating injury at these sites through kind of a new lens might be a worthwhile exercise.
Another comment came into the chat from David, and he notes, I would suggest that statistical significance is no longer tied directly to p-values, and gives the example of the Bayesian approaches.
It's a good observation, but within statistics, significance is by convention tied to the p-values. It's sort of like almost a tautology. The definition of significance is tied to that, but it is, I could see also legally, perhaps it's a different definition than the statistical one. But Barnett, do you have any comments on that?
Not on that one, but I think one thing that you said during the presentation, and maybe I touched on, big decisions should not be made just on a p-value. And I think that is really well known in natural resource damage assessment in business and in many other fields. It's one piece of information or datum that, contributes to the decision.
And Barnett, you actually transitioned nicely to Jeff's question. What do you think is the hardest part for people when changing from a p-value centered approach to a different approach? And personally, I'm young enough and had a progressive advisor statistically where we tried not to use any p-values. And rather than just looking for a magical line in the sand, it's looking at, for me, the context of what's the effect and what's important, rather than just a p-value. But Barnett, you've had experience changing how you think about statistics. So what's your insight to that question?
Oh, gosh. At one time, I thought I knew a great deal about statistics when I came out of school in the first few years in the field, and I was using general linear models, all different types of designs, and really felt like I knew what was going on. But I guess what I've learned over the years is everything changes, certainly in science. Sometimes things have a half-life of about 5 years of interest and examination, and then other information appears or you move on to another area. So I think in some ways you have to kind of give up control, something you're real comfortable with, to really stay contemporary and maybe even gain some better control. That's about it.
This is Joe again. I have a question. And during the presentation, I think it was Barnett had mentioned on some of the graphs, you need a large sample size. And I think you gave some examples of at least, 50 individuals of 3 species. Those aren't exact, but can you describe, is that outlined in the paper that that we referenced at the beginning? Is that where can folks go to learn more about appropriate sample sizes?
Sure. Well, I think one area I was talking about was toxicity, or not. I was talking about were reference intervals used in human medicine, veterinary medicine, and in other fields. And what they like or require is data from about 20 individuals, mixed sex, half males, half females, to come up with a reference interval. So that's, pretty doable. The problem that we face in our field is finding individuals that have not been exposed to contaminants. And, you can sometimes do that with a captive colony or an aquarium or whatever, but that's tricky. I think I gave also the example of the toxicity reference values and what is required to really generate a real robust toxicity reference value. And there I gave the examples of 3 different species within a vertebrate class representing 2 orders, and all these lowest observed adverse effect levels and no observed adverse effect levels. And for that, the bar is very high. It's very difficult to do. I would think for certain types of chemicals, some metals, there may be and there is enough data to do that kind of thing. For persistent organic pollutants, and even certainly new compounds, new chemicals, it's more difficult to get that kind of information to really fulfill all those criteria. And sometimes you just can't do it. You do the best you can, and then you report the uncertainty and what is missing, and perhaps use the word, it's a preliminary estimate or a preliminary value.
Thanks, Barnett. Thea asks, could you talk more about how to deal with non-detects?
Well, I'll say a little bit, and it's kind of tongue-in-cheek, Thea, and I think I know who you are. And I would say the best way to deal with them is look at the book published by Dennis Helsel in 2005. It's in the reference list we provided. I've interacted a little bit with Dennis. He's retired from the USGS. I believe he's still operating. He has a little consulting firm. I gave what I have used in the past, which is kind of a KISS, keep it simple, stupid method, where what you present, it's a range for the mean. It's not a single value. And certainly an easy approach for looking at differences would be using non-parametric methods. Again, you'll end up with a p-value, but that's another approach. So I'd suggest take a look at Helsel. You can actually probably still contact him. You can talk to me. There are a lot of different ways to handle it, and it's very central to our field.
I would add, my first response was going to be the same as Barnett's, where look at that book. Joe also commented that Dennis is retiring, or she's heard from him, but his book is good. And there's also, I use R for my statistics. So there's a couple of R packages for non-detects as well. And Dennis wrote one. There's also another one, and I don't remember their names. I just search for them. And that's how I personally deal with it as well. I'll also say that this is an open statistical research topic, so there are new methods being developed as well. So depending upon how important it is for you, the existing R packages might work, or you might need to do your own statistical research. So, or find a collaborator to do it. So it's always statistics and modeling. It's like the science, how much is enough? And usually what's off the shelf is enough for me with statistical stuff. And if it's not, that's when I start to create my own products and things. So Thea, does that help you?
Hi, it sort of. It's just, I did actually already open the book just because I was looking at your references, and it's it's a long treatise on the subject. So yeah. But else anyway, yes, it's a very central issue to the work that we do. And I know in the past, lots of people had referenced or paid attention to Dennis's practical stats. I don't know if it was a newsletter, I can't remember now, and I don't know if that's archived online, but that might have some, I don't want to say less dense, but shorter descriptions of some of these issues as well, and possible solutions.
Mm-hmm.
His R package, I think, is well documented. So you could, one way I teach myself new code is just to get the example working, and with the R package, and then I replace their data with my own data.
And is that R regression? Is that basically or the R package approach? Is that basically doing a censored regression? Is that, because I'm looking in your paper right now, and I've not done a censored regression before, but is that basically what the R code is doing?
I forget that there's two packages. One is called censored regression, or sensreg, and there's also a Dennis's package. I'm not sure if his is that one or it's a different one, but yeah, like the one we used, it was. It's not that hard to run the code if you know R well. It's just understanding what the model outputs mean, both statistically and biologically, where things become more important.
Right, for sure. Thanks. Looks like Katie, thanks for dropping in that practical stats web link. And other folks have some more information there. This may not be a question that Barnett or Richie could respond to, but it just seems to me that from a NRDA case perspective, maybe we have forgotten in the past, and maybe it's just not NRDA, but our science projects in general, that having a statistician as part of the team early on could be very beneficial versus, I know even with my work, I'm guilty of some of your errors that you noted on the slides that, oh, I took some stats at the graduate level and I learned enough in my eco talks and that's enough, but maybe it actually isn't if we want to be more robust in this approach.
Barnett, do you have any comments?
I agree with what Joe said.
Yeah, and I would agree as well. When I'm reviewing statistical proposals, it's not like a bad peer review where somebody's out to grill you. It's a collaborative where we want the study to go well. So it's discussing what's important. It's sort of like animal care and use, where it's helping you make sure you're using the animals appropriately in things. Statistics is also getting the statistics involved. Just make sure you have like a large enough sample size for your question and some things. So another point I would add is if things are important enough, you can also either do like the power analysis ahead of time, where biomedical studies will do that to see what's their sample size. Also, working with Fish and Wildlife for the environmental DNA monitoring of invasive carp, we've done simulation studies before to see how many samples they need. And if it's like a multi-million-dollar sampling project, then it's probably worth the time to do a $10,000 simulation ahead of time to see how many samples you need. And Kristen also notes, and involving just like your comment, and talks about just what I said. What's your required sample size, power, and other things. So it's looking at I think, Joe, it ties into some budgets. I know NRDA can sometimes fund large science projects. So before you spend $3 million, I don't know, X million, whatever it is, I'm making up numbers. It might be worth it to spend a small amount just to make sure the statistics is appropriate. Either the review, or if it's important enough, you simulate the data ahead of time to see what do you think is the method appropriate for your question. And it might be iterative. You do a lab or a pilot field study and get some base data, say, we think this is how variable the data is. We think this is what the difference is. If we roll this out to a landscape, let's say it's contamination, I don't know from what, you might do a pilot site at a known hot site and say, this is what we think the contamination level will be. This is how much variability there is. And then to roll this out across multiple sites on a landscape, how many samples do we need to spend? So that just elaborates of how you can, it's an iterative process. You collect data and then you see what you need for bigger level inferences. And
so Richie, that's great advice. But what if your study's already underway? What if you missed that initial boat of having your statistician join you early on? Is there a point where you're just not going to be able to reevaluate, or would you recommend, even if you're in the middle of a study, to try to do some reevaluation of the statistics?
Getting the statistician involved as early as possible is really good, even if it's part way. It might be the worst case is that your experiment's not salvageable, and then it's better to learn that halfway through, and then you can maybe change something or scrap it then rather than continuing all the way through. Something where people are like really good at experimental design, somebody like Barnett who has a lot of experience, or like he can, even if Barnett doesn't know his exact statistics, he can set up, and Barnett does do this, but he can set up a good study that a statistician can salvage later. The opposite's not possible, though, where like a bad study can't be salvaged necessarily by good statistics. So to answer your question, partway is better than never. And I've been in that situation. Just when I joined my center, I joined projects at the end where people are like wrapping it up or partway through and have confusing results.
Yeah, this is Barnett. I have a comment. A lot of the NRDA work is field studies that are multi-year field studies, and data are generated after year, and halfway through a study may be a great way to take a look at the data if things aren't designed so well, because it might give you an idea of how many more years or how many more seasons of data you need to collect to salvage the study. And I think that's where a statistician could really come in and be a big help in the middle of a study.
Great. Yeah, Jeremy's comment sort of alludes to that as well. He notes some EPA has some guidance and putting the effort in at the beginning could pay dividends later. So we only have a minute or two left. Any closing comments that either of you would like to make, Richie or Barnett?
Thank you for the opportunity to speak today. It's been good to get to know this group better.
Ditto.
Well, thank you both. I'll try to drop your all's emails into the chat, or if you could each do that, that'd be fantastic. I got to run to another call, but I know we talked before, and you're willing to have people contact you afterwards. So if you didn't get your question answered, or if you think of one after, please feel free to reach out to Richie or Barnett. So thank you all once again, and hope everyone has a great afternoon.
Take care.
Bye.